Good day, everyone, and welcome to another screencast here in Immunology and Serology. I am your professor, Dr. Supache Basit, and for today's meeting, we shall be discussing cytokines. For this session, of course, I'll be introducing to you cytokines, and then what are the different cytokines that are involved in innate immune response. Cytokines involved in adaptive immune response, the Th17 cytokines, and then discussions also, brief discussions on hematopoietic growth factors, cytokines and anti-cytokines therapies, and what are some of the clinical assays for cytokines. Okay, so moving forward, when we say cytokines, cytokines are defined as small soluble proteins. Small that may be, but they still play a very important role in our immune system for the very purpose or for the very fact that these cytokines are able to regulate our innate immunity. And of course, they are also important as as part of our adaptive response to infection. So cytokines are actually being induced whenever there are certain stimuli. And then these stimuli could be in the form of bacterial lipopolysaccharides, the LPS, which is found most of the time in gram-negative bacteria cell wall, flagellin, a protein of the flagella for motile bacteria, other bacterial products, and the ligation of cell adhesion molecules. Okay, so there are different ways in which the cytokines act. The first is known as the synergy. So when we say synergy, cytokines act together. So the presence of one substance, as you can see here. So as you can see in the diagram, B alone, the target cell response is very low. But if we combine it with A, the response becomes very high. Because if you're comparing A alone and B alone, the target cell responses are very low. So that's what you call synergy. On the other hand, when we say pleiotropy, one cytokine has many. Okay, one cytokine has many action, and when we say redundancy, different cytokines have the same function. So it's different from pleiotropy because in pleiotropy we're talking about one cytokine only. But in redundancy, we're talking about several cytokines, but with the same function. Okay, antagonism is the exact opposite of synergy. Here, in antagonism, cytokines counteract each other. As you can see here, C alone, the target cell response is very high. Okay, but when you combine C with A, then the target response becomes very low. So that's what you call antagonism. Okay. Cytokines, the in vivo effects of cytokines expressions are actually visible on leukocytes and other cells because they are not really visible, but they are being manifested on leukocytes and other cells because they are important for regulation of growth, differentiation. So in the context of hematopoiesis, when we say differentiation, this may pertain, of course, to the maturation, differentiation and maturation, gene expression. Okay. So cytokines will also exert effect through the following. So when we say autocrine, okay, autocrine stimulation. So the same cytokine, okay, for example, the macrophage here produces interleukin 1 beta. So the cytokine that is produced by this macrophage or by this antigen presenting cell will have an effect, okay, to the same cell. So it will stimulate macrophage to produce more cytokines, to become more active. Okay. So when we say paracrine stimulation, in paracrine, what I've said in paracrine stimulation, as you can see here, the cytokine acts on the nearby cells. So as you can see in the illustration on figure 6-1, An example here is that the interleukin 1 beta stimulate the T helper cells, the Th2 cells here. Okay, it also activates another cell here, neutrophils, and it also the interleukin 1 beta was also able to depolarize the neurons. Okay, so the other one is systemic or the endocrine function. So for the systemic or the endocrine function, cytokine travels through blood vessels, okay, to distant cells. So as you can see here, the interleukin 1 beta stimulates acute phase protein production in the liver and also fever induction in the hypothalamus via the blood vessel. Okay, so that's what you call the endocrine activity. Cytokines have also multiple actions. So for the very reason that okay, there is a widespread distribution of receptors on many cell types. So that's the reason why they have multiple actions. Okay, and another thing is that they have this ability to alter expression of numerous genes. Okay, cytokines may also share properties and receptors, and they may have overlapping effects. Okay, so that's the reason why we'll have the principle of pleiotropy, we'll have the principle of redundancy. And cytokines can also determine whether the host will be able to mount an effective defense. Let's say example against a certain pathogen. Okay, so there are many cytokine families. Okay, so they may be classified as the TNFs, the tumor necrosis factors, the interferons, the chemokines, the TGFs or the transforming growth factors, and the colony stimulating factors. Okay, so let's first talk about the cytokines in the innate immune response. Okay, so these cytokines are involved in triggering recruitment of effector cells. So examples of these cells are the interleukin 1, the tumor necrosis factor alpha, the interleukin 6, the chemokines, the transforming growth factor beta, the alpha, and the beta interferons. So let's talk about first interleukin 1. So interleukin 1 acts as an endogenous pyrogen. It's like having an internal thermostat that can actually shoot up your temperature, because interleukin 1 can induce fever in acute phase response through its actions on the hypothalamus. Remember the interleukin 1 beta, okay, which can travel to the blood through blood vessel and goes directly to your brain. Okay, it will tell the hypothalamus that it's about time to raise the temperature of the body. So it induces the production of the vascular cell adhesion molecules and the presence of other cytokines such as the chemokines and interleukin 6. So some examples of interleukin 1 are the interleukin 1 alpha, interleukin 1 beta, and the interleukin 1 RA, or the receptor antagonist. So both interleukin 1 alpha and beta may be considered as pro-inflammatory cytokines, which means that they mediate in inflammatory response. Okay, so they are produced primarily by monocytes, macrophages, and dendritic cells. So what are the common denominators of these cells? These cells are all considered as the APCs, or the antigen-presenting cells. So interleukin 1 beta, on the other hand, is responsible for most of the systemic activity that is attributed to interleukin 1, such as the presence of fever, whenever our phagocytes are being activated, and the production of acute phase protein such as the C-reactive protein in cases of inflammation. Hence, they are considered to be as the pro-inflammatory cytokines. So, interleukin 1 RA, or the receptor antagonist, is produced by monocytes and macrophages. So RA means it is a receptor antagonist because it actually acts as an antagonist to interleukin 1 itself. How? By blocking the receptor of interleukin 1. Therefore, if the receptor of interleukin 1 has already been blocked, then it will limit the availability of the receptor for interleukin 1. Okay? So Interleukin 1 RA is actually produced as maybe perhaps as a maybe not perhaps but is produced so that there would be some regulatory mechanism for the production of the for the actions of the interleukin 1 in particular. Okay, so let's talk about the alpha tumor necrosis factors, one of the most prominent cytokines under the TNF family. So it exists in either membrane bound, or if they are free in the solution, or free, which means that they are not attached. So we call it the soluble form. So the alpha TNF may cause vasodilation. So vasodilation means that the blood vessels becomes dilated, and in doing so, it will increase the extravasation of blood. No, it will increase it. The blood vessel becomes more permeable. So vasopermeability. So more fluids will come inside the blood vessel. So that may also trigger the presence of edema, for example. So it is triggered also by the presence of lipopolysaccharide, LPS. Okay, this is one of the major virulence factor found in the cell wall of gram-negative bacteria. So the alpha tumor necrosis factor is activated by monocytes and macrophages. Okay, again, antigen-presenting cells. So it can activate T cells through its ability to induce expression on MHC class II molecules, okay, vascular adhesion molecules, chemokines in similar manner with that of the interleukin 1. Okay, so it can activate T cells. Okay, so interleukin 6 is the one that is a single protein produced by both lymphoid and non-lymphoid cell. So again, interleukin 6 is actually a part of cytokine cascade. So cytokine cascade because there is a series of cytokine being produced or released in response to lipopolysaccharide. It is pleiotropic and plays a role also in acute phase reaction. So it is triggered by the presence of interleukin 1. Okay? So interleukin 6 stimulates B cells to proliferate and differentiate into plasma cells. So it will induce CD4 positive T cells to produce greater quantities of both pro and anti-inflammatory cytokines. Previous discussion, so we identified CD4 positive cells as the T helper cells. Okay, so it stimulates B cells to proliferate and differentiate into plasma cells, and as we all know, plasma cells are the ones responsible for the production of the antibodies. Now let's talk about chemokines. Okay, we're still under the topic cytokines present in innate immune response or natural response. So chemokines will enhance the motility, that's one, and promote migration of many WBCs toward the source of the chemokine or toward the source of stimuli, chemical stimuli. So we call it chemotaxis. So if WBCs are attracted toward a certain chemical stimuli, we call it chemotaxis. And we can classify chemokines according to the location of the N-terminal cysteine residues. So in between C, that's alpha, so CXC. Two consecutive N-terminal cysteine residues, we call it beta. A single cysteine, C. And then you have cysteine followed by CX3C. So you have a cysteine and then another X and then another 3 more cysteine. So that's why CX3C. Okay, so that's the classification of chemokines. Do you know that T helper cells have chemokine receptors, okay, on their surface? Okay, so examples are CCR5 and then CXCR4. CXCR4. So these are the chemokine receptors at the surface of of the T helper cells. And HIV binds to CD4 and chemokine co-receptors. Okay, so there are some researchers that they are developing drugs that will somehow will cause endocytosis of CCR5 and CXCR4. So that way, HIV, the virus, will not be able to penetrate CD4. So yeah, so that's the so that's one of the researches that I've read about chemokines. Okay, chemokines are involved in the initiation and development of inflammatory responses in numerous disease processes. So again, chemokines, just like other cytokines, may be involved in inflammatory response. So these chemokines will combine with cell adhesion molecules, and it will facilitate the extravasation of leukocytes into the tissues in areas of inflammation. We simply call it diapedesis, wherein leukocytes will actually travel out of the blood vessel in order for them to immediately go to the area where there is an inflammation. Okay, and then let's talk about the TGF beta, tissue growth factor beta, composed of three isoforms, beta 1, beta 2, and beta 3. So, these particular cytokines will induce anti-proliferative activity in many cell types. So, anti-proliferation. So, proliferation means multiplication of a certain cell. Anti-proliferation is very important, particularly if you want to control unwanted cells such as cancer cells. Okay? So, which means that the TGF-beta can regulate cell growth, differentiation, even the programmed cell death such as apoptosis, migration, and the inflammatory response. So, this one is very important because it will help down-regulate inflammatory response, especially when inflammation is no longer needed. Maybe because we're able to localize already the infection, or tissue healing or tissue repair has already been taken place. So there's no need anymore for inflammatory response. So that's the time that TGF beta should play a very important regulatory function. Okay. So the TGF beta has a regulatory function because it can inhibit activation of macrophages and the growth of different somatic cell types. And having said that, it may also function as an anti-inflammatory factor for many mature T cells. So in activated T cells, it inhibits proliferation. And it may function as an autocrine regulator to limit the expansion of activated cells. So, even in politics, there is a need for us to have a fiscalizer, something that will regulate, something that will not really fiscalizer, but something that will referee the overactivity of a certain person. So we can actually relate that, or we can actually have an analogy with that to that of the TGF beta. So a cytokine that will control the activity of a certain cell. Important for homeostatic mechanism. Okay, so type 1 interferons could either be alpha interferon and beta interferon. Okay, it's very important because it's like a natural antiviral substance inside our body, because these interferons may interfere with viral replication. So they are being produced by dendritic cells, and it will induce production of proteins and pathways that interfere with viral replication and cell division. Okay? So it activates also natural killer cells, the large granular lymphocytes, and as we all know, natural killer cells will destroy target cells. And when we say target cells, these cells are either cancer cells or somatic cells that have been virally infected. So type 1 interferons will enhance the expression of MHC class 1 proteins. MHC class 1s are actually being expressed by all somatic nucleated cells of our body. So they are active against certain malignancies and other inflammatory processes. So let's talk about cytokines present in adaptive immune response or specific immunity. So these cytokines are mainly secreted by T cells, particularly that of the T helper cells. So even if they are being secreted primarily by T helper cells, it may also affect the function, not only that of the T cells, but the B cell function as well. So it is more, this affect T cells and B cell function more directly than cytokines involved in innate immune response, because cytokines involved in innate immune response more or less would have a general non-specific response, okay, not cell-specific, okay, because these cytokines will directly affect specifically the T cells and B cell functions. So there are actually three main subclasses of CD4 T positive cells. These are the Th1, Th2, and the Tregs, or the T regulatory cells. So let's talk about these in detail. So remember that when the T cell receptor, or the TCR, found on the surface of CD4 or the T helper cell captures an antigen, that particular T cell will undergo clonal expansion. Meaning, if a certain T helper cell capture an antigen, that antigen will actually, that particular T cell will actually proliferate. So that T cells that have proliferated may be differentiated and mature into Th1, Th2, or Treg. So there are three possible choices. So there are three lineages. And this differentiation is actually influenced by the spectrum of cytokines expressed in the initial response, as shown here. Okay, so this particular illustration will show the subpopulation of cells. So you have here the antigen presenting cells, which could actually encounter different pathogens such as bacteria, viruses, fungi, extracellular pathogens, okay, parasites. So depending upon the pathogen encountered, okay, the antigen presenting cells, which could be a macrophage, okay, will secrete a specific combination of polarizing cytokines that direct the naive T cells to differentiate into, as what I told you, different lineages. Naive T cells, because when we say naive, these T cells have not encountered any pathogens yet. So it's the first time that they're able to encounter the pathogen. So this naive T helper cell will no longer be naive when it encounters pathogen. So it will undergo clonal expansion. Okay? So it will be differentiated into specialized subsets. So each T cell subpopulation exerts different immune functions. So they will actually release the release of effector cytokines will coordinate an appropriate immune response directed against these pathogens that the antigen presenting cells have encountered and presented to the naive T helper cells. So as you can see, if the subsets are the Th1 and Th17, so these particular subsets will be involved in cell-mediated immunity. So the effector cells are cytotoxic T cells and macrophages. Okay, if Th2 is involved, so possible interleukins that will be produced are interleukin 4, 5, and 13. Effector cells are mast cells, B cells, and eosinophils. And this will be primarily be involved in humoral immunity. Okay, so let's talk about the different adaptive immune response cytokines. So we have the Th1 cytokines. So we have the gamma interferon. So it affects RNA expression levels of more than 200 genes. Wow. So it stimulates antigen presentation by class 1 and class 2 MHC molecule. So the thing is, gamma interferon will activate macrophages so that it will stimulate phagocytic and cytotoxic abilities. So as we all know, if macrophages were able to phagocytize something as an antigen-presenting cells, this particular macrophage will present it to the CD4 positive T cells. Okay, so Gamma interferon will regulate the action of CD4 positive Th cells, CD8 positive cytotoxic lymphocytes, and even that of the natural killer cells or the large granular lymphocytes. Okay, still under the Th1 cytokines, we have the interleukin 2. So interleukin 2 is otherwise known as the T cell growth factor. Okay, so it drives the growth and differentiation of T and B cells. It also induces the lytic activity of natural killer cells, the ability of the natural killer cells of the large granular lymphocyte to cause lysis of target cells. It can activate proliferation of Th2 cells and help to generate IgG1 and IgE producing cells. And then we also have the interleukin 4. So interleukin 4 is very important because it helps drive antibody response in various diseases such as allergies, autoimmune diseases, and parasitic infections. Okay, so interleukin 4, the receptor is expressed on lymphocytes and various non-hematopoietic cell types. And then the interleukin 10, and this one has an anti-inflammatory and suppressive effects on Th1 cells. Okay, so anti-inflammatory. So when the process of inflammation is no longer needed. So it is produced by monocytes and macrophages, CD8 positive T cells, Th2, CD4 positive T cells. Okay, so interleukin 10 will inhibit antigen presentation by macrophages and dendritic cells. So it inhibits antigen presentation, so more of a regulatory function as well. So it serves as an antagonist to gamma interferon to downregulate the immune response. Again, interleukin 10 has more of a suppressive and or regulatory function. Tregs. So Tregs are cell populations that have the following marker: CD4 positive, CD25 positive T cells. So Tregs will help peripheral tolerance to various self-antigen, allergen, tumor antigen, transplant antigen, and infectious agents. So peripheral tolerance is very important for let's say prevention of the occurrence of autoimmune diseases, and of course it's also very important for the prevention of rejection of transplant antigen. That's very good. Peripheral tolerance is also important so that will not exaggeratedly react to allergens. Okay, and however, it may not be good if we'll be able to tolerate figuratively and literally tumor antigens and infectious agents. Okay, so it produced the TGF-beta that has the ability to downregulate or suppress the activity of other T cells. So Tregs will induce interleukin 10, the tumor growth factor beta expression in adaptive TR1 cells in the peripheral circulation. So T cell suppressions occur through interleukin 10 inhibition of pro-inflammatory cytokines. Okay, it also occur T cell suppression occur also through inhibition of co-stimulatory molecule expression on antigen presenting cells. So the first bullet or the first sub-bullet will tell us that interleukin 10 indeed has a very important regulatory function. Okay, the tumor growth factor beta will downregulate the function of antigen presenting cells such as that of the macrophages and block the proliferation and cytokine production by CD4 positive T cells. So the immune response is downregulated, and that's very important because chronic inflammation could also be prevented. You know what? I have a story to tell you. During the 1918 pandemic, majority of people who died from that H1N1 flu are young adults, okay, or adults or people who are at their prime, who at that time whose immune system is actually very strong. And you might be wondering, their immune system is strong. How come how come the mortality rates population actually belong to their age group? Primarily because they have a strong immune system, and the presence of H1N1 causes cytokine shower. So just imagine. So when you say cytokine shower, literally your immune system produces, okay, in exaggerated manner, different cytokines, and some of these cytokines induces inflammation. So we call it the pro-inflammatory cytokines. So we do not want our body to have cytokine shower because it will also affect our system as well. So that's the reason why there is also a need for us to have cytokines that will somehow cause homeostatic balance, so that we do not want cytokine shower to occur in uncontrolled fashion. Okay? So let's talk about the Th17 cytokines in innate and adaptive immunity. So they have a role in host defense against bacterial and fungal infections at mucosal surfaces. Okay? So Th17. So it's very important for innate and adaptive immunity. Mucosal surfaces. Mucosal immunity is very important because when you say mucosal immunity, this may be considered as the first line of body defense as well, before these pathogens will literally invade the entire body system. So the reason why we call it Th17 cytokines, obviously, because this involves Th17 cells. Okay, Th17 cells secreting interleukin-17 family of cytokines to promote continuous recruitment of neutrophils. So this will promote release of antimicrobial peptides. So when dysregulated, okay, this particular scenario will have been implicated in pathogenesis of multiple inflammatory diseases and several autoimmune disease conditions. So let's talk about the colony stimulating factors. So this includes interleukin-3, erythropoietin, granulocyte colony stimulating factor, macrophage colony stimulating factors, the granulocyte macrophage colony stimulating factors complex. So all of these cytokines are involved in differentiation of blood cells in the process of hematopoiesis, which happens in the bone marrow in response to inflammatory cytokines such as interleukin-1. So because of these colony stimulating factors, our blood cells will actually mature and becomes pale. One of the most important cytokines is the EPO or erythropoietin, which is primarily produced in the kidneys. So EPO's production is actually induced by a condition known as hypoxia or the condition of low oxygen tension. So EPO therefore regulates RBC production in the bone marrow, as again proliferation of RBC is induced by EPO. So whenever there is an EPO, therefore will improve oxygenation of the tissues because you have more RBCs, and eventually will switch off EPO production if it is no longer needed. Okay, because the proliferation of RBC means there's more hemoglobin, and then it will cause oxygenation of the tissue. Homeostatic mechanism is very important. Okay, so let's talk about various therapies and approaches. Okay, so sometimes we do not want cytokines to be exaggerated with reduced. So that's why there are some drugs that may disrupt the interaction between cytokines and recombinant receptors, such as the infliximab. Some will utilize monoclonal antibodies that may serve as cytokine agonists. Some may utilize hybrid proteins containing cytokine receptor binding sites attached to immunoglobulin constant regions to block cytokine activity, especially if that cytokine is pro-inflammatory and we do not want, again, to have a chronic inflammation. So that is an example of the example of the action of drug known as the etanercept. Some will even block interleukin-17 function, such as the ixekizumab. Okay, so how do we say for cytokines, we can do the ELISpot ELISpot assay, which is actually an ELISA technique, enzyme immunosorbent assay technique on in vitro activated peripheral WBCs. Some are actually using the multiplex ELISA. So multiplex means that it can actually detect simultaneously about 12 to 25 pro and anti-inflammatory cytokines in one reactions. And then some would actually utilize the microbead assays, which will allow for the simultaneous detection of, again, multiple cytokines in a single tube. And that's the end of our discussion for cytokines. I hope you learned something today. If you have any question, do not hesitate to message me. And of course, in this time of uncertainty, I'm wishing everyone to to be safe, stay safe, and God bless everyone. See you around.